growth of microbes is something that we definitely have to keep in check and keep under control. And that's not just on surfaces in our environment, but also on our body. So we do have a number of antimicrobial drugs and different approaches that are used to control microbe growth. And that's both to control pathogenic microbes and also to ensure that we don't get overgrowth of microbes that would typically not be pathogenic. If we talk about drugs in general, just that term when it's used generically refers to any chemical that's capable of affecting physiology in some way, so in some manner. And that is a very wide definition. That includes some of the foods that we eat, things like chocolate or coffee, things that have caffeine, because that does alter our physiology. Tobacco products would fall into that category as well. But if we're talking about things that we would use to specifically fight diseases, we use the term chemotherapeutic agents. And that would be things that could fight genetic disorders or things like cancer. Those are things that are not really caused by microbes or caused by infections. One category of that, though, is going to be antimicrobial agents. And antimicrobial agents are antimicrobe. So these are going to be things that specifically attack pathogens. So they will be helped to treat infections that we do get. If we talk a little bit now about the history of antimicrobial drugs, we have seen a huge amount of development of this in just the last century. In the early 1900s, 1/3 of children under the age of 5 died from some sort of microbe infection, some type of infectious disease. And that's because we didn't really have ways to treat those microbes. They did understand that microbes were responsible for causing a lot of the problems that they saw, but other than diagnosing them, there wasn't a whole lot that they could do. This timeline right here shows us some of the developments that we did have in antimicrobial agents, so things that we could use to treat infections. And one of the first ones that was discovered, this was just prior to the 1900s, this was an arsenic type compound. And while it was something that did specifically inhibit bacterial cells, arsenic compounds are also toxic to humans. So if we're talking about something that's going to be toxic to humans, that's not going to be a really good drug choice. And so this wasn't something that we could really use for treating infectious diseases. Other developments, we had the discovery of penicillin. That was something that there was some difficulty in producing large quantities of it, so that limited when we could actually use it for treating diseases. So one of the first ones to actually be effectively used was a sulfur type drug. So this is what we recognize as sulfonamide. We recognize it to be one of the first widespread used drugs to treat infectious diseases. There were other ones that were discovered. Many of them in the 1940s were discovered from various soil-dwelling bacteria. So they were things that they did isolate from them. And if we just talk a little bit more about the penicillin example, this is something that was discovered by Alexander Fleming in the 1920s. And he is the first one that discovered that if you do grow this fungi called penicillium, that you had this region right around it where other bacteria were not going to be growing. And so he noticed that there were these antibacterial properties that the penicillium fungus had. And he was able to isolate penicillin, and that is something that we do use now as an antibiotic. Like I said, when he first discovered this, it was difficult to get large quantities of penicillin, so it's not something that was immediately used for treating bacterial infections. Now, when we use the term antibiotic, this is something that is naturally produced. So naturally produced in a fungus, naturally produced in a bacteria or some other organism. If we talk about synthetics, these are things that are completely synthesized, completely manufactured in some type of lab. We do also have what we call semi-synthetics. These are kind of a combination between the two, so they are a modified antibiotic. Now, the mechanisms of antimicrobial agents, if we go through and we talk about what they're actually going to do that's going to help them to be so effective, well, first of all, they need to have selective toxicity. And what we mean by selective toxicity is that they can't kill everything. They can't be so harmful to the host organism because, of course, that's what we're trying to help out. So we need to selectively kill the microbe, but not the host. So this is going to be easiest or work best if you have big differences structurally and metabolically between the microbe and then also the host organism. So if we talk about what the drugs actually do to the pathogens, there's a lot of different ways that they can actually work. One of the things that they do is they inhibit cell wall synthesis. They can also inhibit protein synthesis by targeting specific differences that we have in prokaryotic and eukaryotic ribosomes. They may disrupt unique components of the cytoplasmic membrane. We can have inhibition of general metabolic processes or metabolic pathways that you don't typically find in human beings. And there can also be inhibition of nucleic acid synthesis. A sixth one here is that there may be something that they're blocking that affects the ability of the pathogen to recognize its host or maybe specifically attached to its host. So any of these different methods here could be used by antimicrobial drugs, and exactly what the target is or exactly what it's actually doing is going to depend on the drug that we're actually using. If we talk about the inhibition of cell wall synthesis, first thing to remember is what type of organisms actually have a cell wall. So this is going to be something that we find in pathogenic bacteria, and we also find it around fungal cells. And bacterial cell walls, if we focus on those specifically, these are composed of peptidoglycan. Peptidoglycan is going to be some sugars and also some peptide components, and these are going to be all cross-linked together, and you can see that in this image here. So notice that when the cell walls are composed, you do get these little cross bridges that form between some of the subunits. And every time new subunits are incorporated, those new cross bridges have to be formed. Well, one of the ways that some antimicrobial drugs work is that they actually prevent this cross-linking from taking place. So we can have bacterial cells that make kind of the initial components of the cell wall, but they can't finish compiling those and hooking them together. So here's a look at some of the different drugs that are able to do this. Penicillins, they do function in this way. They prevent that cross-linking from taking place. And if you look at the image here, you can see that any new cell walls that are going to be formed, we're not able to put those cross-links. So see that we're missing them here, and that's going to make for a cell wall that is a lot less capable of withstanding certain osmotic pressures. So if there is a situation where fluids try to move into the cell, there's a very real possibility that that cell is going to end up rupturing, which would end up with a dead cell. So that's one way that these drugs can work. So ends up with a cell wall that bursts or ruptures when under osmotic pressure. Now, some semi-synthetic forms of this particular compound, they're going to be more stable in acidic environments, and also they're going to be more readily absorbed. They're less susceptible to inactivation by bacterial enzymes, and so that's one of the reasons why we do a lot of times use semi-synthetics. They do have some enhanced capacities that the natural forms do not have. If we look at what some other drugs can do with regards to the bacterial cell wall, we do have some that interrupt some alanine-alanine bridges within the cell wall. So this is again kind of linking components together. We have some cases where there's actually a blocking of the individual subunits that are being made to compose the cell wall, basically blocking the transport of those from the cytoplasm to the exterior where they need to be incorporated. So with all of these methods of blocking cell wall synthesis, one important thing to keep in mind is that all of these affect new cell walls that would be formed. They're not going to do anything for already established cell walls. So these are primarily going to affect new cells that are being produced. If we talk now about fungal cell walls do have a different component in them. They're not going to be composed of peptidoglycan like our bacterial cells, and instead these actually have a very unique sugar component. This is the glucan that you see right here, and since it is unique to fungal cells, this is a really good target for some antifungal drugs. If we look at another way that antimicrobial agents can function, another thing that they can do is they can inhibit protein synthesis. And proteins have such important roles inside any cell that if you are able to actually turn off the synthesis, the making of new proteins, that has a dramatic effect on organisms. So one of the ways that these actually work is that they have to specifically target some differences that we do have between bacterial ribosomes and eukaryotic ribosomes. They are really similar structures, but there are some unique characteristics, and you can see there's a few shape differences when we are looking at the ribosomes. So the ribosomes in mitochondria, they can be very similar to the bacterial ribosomes. So that is one thing that we have to be really careful about when we are looking at these types of drugs, because we definitely don't want to inhibit protein synthesis in the host cells, so in the human cells. If we talk about how these actually function, we do have to think about how the proteins are actually going to be made. And this process, translation, which is what we're looking at here, it takes an mRNA sequence, which is the template, and then the ribosome is going to read through that. And as it reads through those instructions, it's going to hook together amino acids that are being delivered by tRNA molecules. So here's 6 different ways that these protein synthesis inhibitors can function. One of the ways that they can function is that they do alter the overall structure and the shape of the mRNA. And when this happens, this causes the ribosome to actually misread the template. And if it misreads the template, it misreads the instructions, and now it's not going to make the proteins the correct way. So we're probably going to end up with some non-functional proteins inside the cell, which would inhibit that bacteria from surviving. Another way that they can work is they can actually block the reading of the mRNA template. And the way that they do that is they block the tRNAs from actually attaching to the mRNA, and that's an important step that we do have in translation. Another process can be that the peptide bonds that need to form between the amino acids, they're not able to form. And that's something that the ribosome really needs to do, is it needs to fit those 2 amino acids together. So if we prevent that, we would really be preventing all protein synthesis. Another way is that they prevent the ribosome from continuing to move along, continuing to move forward on the template molecule. If you look in E, figure E here, we have an antisense nucleic acid. And what the antisense nucleic acid means is that this one runs anti-parallel. It's an opposite or complementary sequence to a molecule, an mRNA molecule that we have. And if you have that mRNA and it's bound by an antisense molecule, the ribosome's not going to be able to actually bind to the template. And then the last one that we see right here is that there can be a blocking of the overall assembly of the initiation complex. If a drug certainly binds to the template, it can affect that. And if we never are able to form that initiation complex, then certainly we're going to prevent the whole process of translation from taking place. So all of these that we're seeing on here are just different examples of how certain antimicrobial agents are going to block the overall function and activity of the ribosome. And if they do that, they are going to prevent that cell from producing proteins. The disruption of the cytoplasmic membranes is a big one. So we have a number of drugs that do affect that. Some antibacterial and antifungal drugs, what they do is they actually create pores or channels in the membrane. And we have to keep in mind that the membrane is crucial for regulating what gets to go into the cell and what exits. And so what these are doing is they are creating these pores that do not control what goes in and out. Pretty much anything can now flow in and out of the membrane, and that would be something that's deadly to a cell. So here we have an example of one specific drug that does do that. This drug, it interacts actually with some other components in the bacterial membrane. And here we have an example of how it does interact with this particular egosterol. And with this one, especially if we're looking in fungal cells, you can see that you get this pore now in the membrane, which means anything can basically flow back and forth through this pore unregulated. So again, that's not a good thing for the cell. Also, some drugs are able to affect the overall membrane transport. So they might inhibit the ability of the membrane to pass things across, so from one side to the other. There can be just an overall inhibition of metabolic pathways. And typically these are going to be antimicrobial agents that are effective in targeting unique pathways. So things that we don't have happening in human beings, but they would be processes that are happening in our pathogen. The example molecules that you see here, some of them are naturally occurring molecules. The first one, this is something that we find inside of bacterial cells, but then the other ones basically mimic that shape. They have the main parts of that molecule, but then there's other things added on to them. And what this does is it actually confuses the cell, and the enzymes end up grabbing those drugs rather than the molecule that they want to actually grab hold of. And this is going to be affecting overall the synthesis of tetrahydrofolic acid, which is a form of folic acid. So this molecule here is typically used to make the THF. And so these other kind of mimics of that molecule, enzymes will grab hold of them, which means that once they grab hold of them, they're not making the THF that we want them to make or that they need to actually make. So here's a look at that process. Normally, this is what we would want to happen over here. That molecule binds in the active site of the enzyme, and then we're going to get the THF that we mentioned here. So that is something that is needed inside of the cells. With the drugs, what happens is they actually bind in the active site of this enzyme, and that means that this one doesn't, and we're not going to get that molecule produced that the cell needs. Now, these are really nice drugs to use when we're talking about bacterial cells because this is not how folic acid is made inside of humans. So with human beings, we basically eat or consume our folic acid, so we're not affected by these same ones. There are some examples where we have antiviral drugs that affect the ability of the virus to shed its coat after it attaches to the host cell. This is a very important step that needs to take place, and you can see that in the image here that you have a viral particle that enters the cell, but once it gets into the cell, it needs to shed what's on the outside so that it can present its genetic information to the cell, the host, because it wants the host to use that information to make some proteins for itself. So if we do inhibit its ability to kind of unclothe, then this virus is not going to be able to replicate inside of the host. There's also some antiviral drugs that inhibit proteases and reverse transcriptases, and those again affect the ability of a virus to replicate. The inhibition of nucleic acid synthesis is another thing that some antimicrobial agents do target. These, however, are a little bit trickier to use. This is because these types of drugs typically do harm the host also. So we have to be very careful because, again, we need selective toxicity. We want to harm the microbe, the pathogen, but we don't want to harm the host cell. There are a lot of what we call analogs, and by analogs, these are things that mimic the overall shape of the normal nucleic acids or the normal nucleotides that we find in our nucleic acids. And on the middle of this diagram, these four in the middle, these are our normal nucleotides. And you can see that all around them, we have things that look fairly similar to them. So what happens is that these get incorporated into the nucleic acid instead of the normal versions, and that affects the way that that piece of DNA or RNA is actually used. So it will ultimately affect replication, it can affect transcription, and it can also affect translation. This is something that oftentimes is used to target viruses. And one of the reasons that viruses are a good choice to use this on is because the DNA polymerases of viruses, they're a lot less choosy, a lot less picky. So they tend to make more mistakes by grabbing these incorrect molecules rather than grabbing the correct ones. And when we're talking about the human DNA polymerases, they tend to do a little bit better job kind of selectively choosing which nucleotides are the correct and proper ones to incorporate. Other things that can affect nucleic acid synthesis, we have some drugs called quinolones. These specifically affect prokaryotic DNA, and they do that by inhibiting DNA gyrase. DNA gyrase is an important enzyme when we are compacting and organizing the DNA in the nucleoid. So they're involved in that twisting of the DNA. And then they also have a really important role when we're talking about the process of DNA replication. So if we have quinolones there, they can inhibit the ability of the prokaryotic cell to replicate its DNA, which means that it's not going to be able to reproduce. The viruses, their attachment to the host cells can be prevented, so some drugs affect that. Anytime we have attachment of a viral particle to a host cell, it's a very specific interaction that has to take place. So there are some receptor proteins that are involved, and we can see those in purple in this picture. And there has to be really this almost lock and key type fit between the receptor proteins and the virus particle. So if we can block that interaction and block that fit between the two, then we can inhibit the ability of the virus to attach to the host cell. And if it is attaching, if we block that attachment, then we block the infection. We call these types of drugs attachment antagonists. So they will be the ones that block the attachment of the virus to the host.